Well, thanks so much, Trang, for the introduction. I just want to thank the whole PacBio team for the invitation to be here. Lindsay, and Jennifer, and Sarah, and Nina, and everybody. It's been a real pleasure. My first time in Vietnam, and last week we were in Singapore as well too. And so it's been a real pleasure to meet some new colleagues and collaborators and to share our experiences. So as Trang mentioned, I'm going to talk today about specifically our long-read HiFi sequencing work over the last 10 years. I just realized last week that it's been almost 10 years since we started collaborating with Jonas and the team. First on amplicons, and then more recently on genome sequencing and panel sequencing. And I'm just going to show this picture really quick here first, not to get too sidetracked, but this was a review that Yao Yang from my team published last year in Genomics in Medicine Open with two other clinical lab directors and showing just really the power of genome sequencing for clinical diagnostics. And without getting into the weeds on these images, these Circos plots, the light blue circle in the middle is the HiFi genome coverage, really highlighting the utility of the HiFi reads for clinically significant genes, which is kind of the driver and the thesis of what we're talking about today. So to start, I'm going to introduce the Clinical Genomics Laboratory at Stanford that I joined the team in 2020. Prior to that, I was at Mount Sinai in New York. And then pivot to our long-read HiFi sequencing applications. I adjusted this from the talk last week. I'm just going to specifically mention the amplicon and genome sequencing application work first, and then get into the work we've been doing in pharmacogenomics at the end, and specifically our collaborative work with PacBio and Twist on the enrichment panel that has been mentioned here before, and how we've recently validated that in our clinical genomics laboratory for actual routine production clinical testing that we're really excited about. So the laboratory was relatively young, I should say. And so we're focused on the germline side of genomic testing. We have a parallel somatic laboratory that we share equipment and staff with. But in terms of our infrastructure, we do clinical genotyping using Agena. We have some NovaSeqs. And I'm really, I feel really fortunate to have such a strong engineering bioinformatics team and a cloud computational infrastructure that really drives all of our workflows. This is just the Illumina workflow, but we actually have a whole new setup now that parallels this for our clinical HiFi computational workload that I'll show a little bit later. And that was because we were thrilled in 2021 when the department supported us after joining Stanford. They knew that our group was really interested in long-read sequencing, and so they invested in a Sequel IIe in the hospital lab. And so, of course, everybody has their own institutions, but the Stanford University and Stanford Health Care are completely different entities. So even though there already was long-read sequencing on the university side, to actually have it in the clinical side was a really big step. And so that's what we're focusing on today. And then also just recently, we got into really rapid clinical genotyping with this device from Genomatics for pharmacogenomic screening for CYP2C19 that I won't get into today. So in terms of the last 4 years in the clinical genomics lab, it's been very busy. And so the whole vision always was to set up clinical genome sequencing as a backbone for diagnostic panels. And so it was really exciting in 2021 to be able to launch our genome backbone diagnostic panels, specifically for inherited cardiovascular disease with you and Ashley in early 21. And then we also launched an exome that Yao Yang designed on a genome backbone. And then every year, I won't go into the details, we're constantly validating something, it seems like, or revalidating something. But in terms of the test development work, we partnered with nephrology in 2022, and then again with preventative cardiology in 23, with some dyslipidemia panels. But more recently, last year, we spent the vast majority of the year drumming up some new test development funding projects. But the big one was our entire interpretation software had to be replaced. And so I won't get into the details here. That's a little off topic. But the main reason why I mentioned this is because of this year, we have a whole new focus. So we've got a bunch of new panels on a genome backbone that we want to bring up. So now in terms of our kind of experience with HiFi sequencing, it did start almost 10 years ago when we were at Mount Sinai. And so we had an RS2 that came pretty early on. And so it was, at that time, we were also really interested in pharmacogenomic sequencing. And this seems to be like the perfect marriage of a technology and an application to us, because in pharmacogenomics, there's really, really important to be able to phase variants and to be able to identify full gene haplotypes. And so Yao had come up with a really basic strategy to basically take full genes as much as we could, depending on their size, barcode, amplify, and submit them for sequencing. And this was prior to all of the great work that Mike has just shown in terms of all the tools that are now available. But back then, Yao was kind of building his own pipelines. But we were just so thrilled because this is a 20,000 base pair, 20 kb fragment that we amplified for STRC, a single sample, and you can really appreciate the beautiful phasing across both alleles across the entire gene. And so for us, the logical first candidate was CYP2D6, and then we heard others kind of mention this gene before. It's one of the most important enzymes involved in drug metabolism variability. It's involved in the metabolism of about a quarter of all medications. And so when we look at sequencing performance across CYP2D6, it's extremely difficult to interrogate. And this is a common theme, I think, of the meeting. But of course, the neighboring pseudogenes are over 97% homologous to the targeted gene. So this is short read sequencing. The top here is exome, and the second row is a targeted short read panel. And I mention that because what we're seeing over on the pseudogenes is really off-target sequencing. And so, of course, the intended target was CYP2D6. So either the capture erroneously pulled down the pseudogene content, or bioinformatically CYP2D6 reads were misaligned to the pseudogenes. But either way, it results in really compromised variant calling for the actual target gene of interest because of all of this off-target sequencing. So we were just blown away when we first started getting into the long-read PacBio sequencing back then, as the precision and accuracy of the long reads in such a complex region was absolutely eye-opening for us. And so it's ever since then, it's just been a real pleasure to continue kind of working through all of these new applications, new targets, and new chemistries, new tools for Mike's team. So it's just been a real pleasure to kind of keep walking through this experience. So after CYP2D6, our next target was SLC6A4, which is another gene for pharmacogenomic association implicated in antidepressant response variability. And that is driven by a variable number of tandem repeats in the promoter that drives expression of the gene. And so this is a well-known association, but it's extremely difficult to interrogate because of the repetitive nature of the promoter, which is shown here. And so these are short reads, again, trying to interrogate this VNTR in the promoter, and it's almost impossible just because it's so repetitive that the short reads can't even map to it. However, using our amplicon strategy shown here on the bottom right, this is a single sample that's heterozygous for the long allele as well as the short allele, they call it, because it has the heterozygous deletion on the bottom. They just beautifully phased all of the relevant variants over the VNTR. So again, another really exciting example of the utility of the long reads, in this case, specifically for PGX. And so this was another candidate that was most recently published from our group. It started in New York, but then ended up in Stanford. And so I believe it was Dr. Tan from Singapore who mentioned it during his talk as well, NUDT15. And so NUDT15, I think, is a really relevant gene to speak at this conference because it's highly involved now, predictor of thiopurine toxicity for children with pediatric leukemia being treated with these thiopurines. And historically, that signal was always driven by TPMT loss of function variants for the last few decades. But it was Jun Yang at St. Jude's who identified a subgroup of patients who still had toxicity when they were treated with the thiopurines, but they were negative for TPMT. And so he was the one who led the GWAS to discover NUDT15 as a second driver of the thiopurine toxicity response. And this is notable because these variants are more commonly enriched in the East Asian population, as mentioned prior speaker from Singapore. And so these findings were quickly translated into updated CPIC recommendations, as well as FarmVar star allele nomenclature ever since those discoveries came out. And then this was a great example for our amplicon strategy as well, because for CYP2D6, the gene is only 5 kb, which is perfect for amplicon. For NUDT15, the gene is only 10 kb. So again, we can amplify that pretty easily. So just to give you, this came out a couple of years ago, but just to give you a little snapshot of the utility of it, this was a Coriell 1,000 genome sample that we actually purchased because it would be a positive control for the star2 allele, which is based on previously reported findings from short read sequencing data. So on the left here, these are the short reads, and then these are the long reads. Of course, we see the beautiful phasing. But interestingly, that star2 is actually overlapping with an insertion deletion variant on 2 different haplotypes. And so the short reads missed the star9 is the takeaway. And of course, star9 is also a loss of function allele. And so these were in trans. And so if we translate that to a phenotype status, we go from an intermediate metabolizer to a poor metabolizer, which has extremely significant clinical consequences for any child who is being treated with thiopurines. They should basically avoid thiopurines if they are a poor metabolizer like this. So really powerful, just again, highlight the utility of the long reads and this amplicon strategy for targets that are amenable to it. And this is just another example as well, underscoring what we already know in terms of the lack of phasing from the short reads and the beautiful haplotype full architecture phasing and the diplotype inference and phenotype inference using the HiFi amplicon reads. So that was the kind of historical backdrop. And so once we got moving in Stanford with the sequel to E, we initially wanted to get our genome, HiFi genome, up and running. And so for that, I just wanted to give a little bit of a context of the sequencing landscape at Stanford as well too. As I mentioned before, we're coming at this from the hospital side. And so in our clinical laboratory, we have the sequel to E, which is being funded through the hospital. But then we also had some pilot funds that I'll mention in a bit to do a small cohort study for undiagnosed disease in pediatrics, who these kids had a negative exome, and so we pivot them to do a HiFi genome. And then of course, what I'll mention a little bit later too, is also our work that's supported by Stanford Catalyst to bring up our enrichment pharmacogenomic panel. But this is not all of the activity, of course, for long-read sequencing. The larger efforts, I would say, have recently employed the Rebio on the university side. And so we collaborate very closely with Stephen and Matt Wheeler and John Bernstein, who are the PIs of the Gregor Consortium, which is essentially like the new undiagnosed disease network in the United States. And so we have a very exciting workflow where for all of our clinical pediatric exome negative patients, we pivot them to Gregor, and if they enroll, we then pursue long-read sequencing, which initially we did on the Sequel, but now is being driven, of course, more appropriately by the Revio. But in terms of standing up the genome sequencing, the very first thing we had to do was our analytical validation work. And so I show these numbers just to give you a sense of how we approach validating any new sequencing technology, whether it's HiFi, short read, or any other type of platform. And so how we approach it is basically standard benchmarking with genome in a bottle samples. But for a better understanding of overall performance, we stratify by different genomic contexts because we know performance is going to differ in different levels of complexity. And then we also stratify by different small variant types because that also influences accuracy and reproducibility. But the really nice thing, and we have hundreds of these tables for all sorts of other kind of analyses that we've done, but the take-home message was that for our small variants, we're seeing well over 99.9% recall and precision across not in the difficult regions, but as well even in the all high confidence regions of the genome in a bottle data sets. And we're happy also to mention that this just came out about 4 days ago in Genome Research, so it's now online if you wanted to see any of the data. And of course, because we also have a lot of experience with our short read data, this is how we validated our short read genome diagnostic panels 4 years ago. We're able to actually compare our data from PacBio to our short read genome data, which is shown here on the left. And so F1 score, F1 score, of course, is like just another accuracy metric that combines recall and precision. But the really cool thing was that everybody talks about depth and cost effectiveness when it comes to sequencing and the appropriate strategy for bringing on these new technologies. And this has been mentioned before by others, but really just validated in our minds in how with the long reads, we actually, our threshold, our benchmark for clinical testing is over 99%, it has to be, and preferably over 99.9%. And so for SNVs, we're hitting that at 15x on the HiFi genome compared to about 30x for the short reads. And then for the indels, we're hitting that at about 25x, again, compared to 30 to 40x on the short read genomes. So again, just another underscoring of how the strategy of bringing on these kind of new chemistries can be looked at in terms of making these decisions on your QC cutoffs and your cost effectiveness approaches. And so we employed that whole strategy to compare the HiFi genome to the short read genome data across many other strata in the genome. And so there's over 100 of them defined by the GA4GH, genome in a bottle, NIST people. They spend their lives, I think, just working specifically on this kind of stuff, but thank heavens they do, because it's been really, really helpful for us. And so this plot up here is essentially the results of the table that I just showed you in the last slide. But if we compare that to the short read results, and this is again stratified by different variant type on the bottom, you can appreciate that the short reads really fall off for the larger indels across multiple different genomic contexts. And this is exacerbated when we look at other regions like GC content regions, as well as tandem repeats, where the long reads are much more stable, much more superior compared to the short reads in terms of their accuracy, particularly in these difficult genomic contexts and difficult variant types. So another piece of analytical validation for any clinical lab, and some others have mentioned this before too, is not just accuracy, but analytical reproducibility. And so this is just showing non-reference genotype concordance across multiple intra-sequencing experiments and inter-repeatability, reproducibility experiments for genome in a bottle reference data. And then the take-home message, though, was that, again, we're hitting 99.9% concordance across multiple replicates of HiFi genome sequencing, and even across difficult regions as well, too, which is extremely important to make sure that the assay is robust enough to implement as a clinical product. And then a third piece on the analytical side that's needed is specimen type validation. And so we were often told that we had to be cautious with saliva DNA for PacBio sequencing, but we were very encouraged. Ever since COVID and telehealth happened 4 years ago, the amount of saliva specimens that we receive now clinically is actually probably 50% of our total specimen type. It's either peripheral blood or saliva. So we really need to have saliva work for all of our diagnostics. And so the nice thing here is that this wasn't even the new saliva kits with the high molecular weight product that I just heard about the other day, but this is actually just using the typical Origin diagnostic kits that most people use. But again, really encouraging to see 99.9% concordance between peripheral blood accuracy and saliva DNA accuracy from the same individuals. So that alone is not enough to launch a clinical test. And so I think there's others here from the clinical kind of space that have talked a little bit about validation, but in the United States, of course, we have a lot of regulatory oversight from CAP and CLIA, and this is just a short list of some of the requirements that we have to achieve. And I mention this because some of them are analytical and some of them are clinical. And so the data that I've been showing you so far is really focused on the analytical side, the sensitivity, the specificity, the accuracy. And so we haven't gotten into yet the clinical validity piece, but I'll talk a little bit more about that when we get to the PGX work. But in terms of our analytical work, we see three kind of products that are needed for our HiFi genome. One of them was that we just published the small variant analytical validation work. And the next two, the reason why I'm so happy to be talking after Mike is because everything Mike and his group do is so critical for our group, is that we have to go now variant type by variant type to validate. And so all of these new callers that Mike has mentioned, Sawfish and the repeat expansions, we're now benchmarking those as well using our similar data sets and our similar strategy of specimen types, reproducibility, accuracy, sensitivity, specificity. But of course, all of these different variant types, either CNVs, structural variants, or the repeats, they have their own nuances, of course, and then different levels of complexity. And so these are the next two work products that we're kind of currently pushing on to kind of wrap up our analytical work for our genome validation data. And so I mentioned that we also are doing a pilot study for genome, HiFi genome work, and this is in collaboration with the Greger group at Stanford. So this is just a snapshot, excuse me, of the pilot that we've been doing for the last few years. So we enrolled about 25, 26 individuals, mostly children, who previously had either a negative or inconclusive exome. Half of them had exome testing internally. The majority of them were trios. In terms of the indication for testing, we had a lot of neurodevelopmental indications, as well as congenital anomalies, other abnormalities as well, too. And so this has been led by Mimi and Rodrigo and Devon, and we've also partnered with GenX for tertiary analyses, who I think were presenting yesterday as well, too. But just to give, this is not complete yet, but just to give a little bit of an update, is that out of all of the 26 in the cohort, we believe we have solved 2 of them now with a novel gene variant finding compared to their previous clinical testing. Several more are being validated by Sanger and scrutinized a little bit further because we have some really strong candidates. And then others are either negative or in progress as well. But then, as I mentioned before, going back to the different variant types, this really is just small variants. And so we still have to work through the important part of addressing the other types of variation that could be behind a lot of these undiagnosed cases. And again, this is a partnership. We love to, we're a clinical laboratory, but we're an academic medical center. And so we really love to work with our colleagues on the undiagnosed disease side, as well as the Gregor side, to actually make these things come to fruition. So with that, I'm going to pivot on the last kind of section here is our work in clinical pharmacogenomics. And so this is something that a field that I've been interested in for about 10 to 15 years for now, back when I was at Mount Sinai. And so part of the excitement to join Stanford 4 years ago was to be closer to my friend and colleague, Terry Klein, who for anybody that does PGX will know who she is because she's the PI of PharmGKB at Stanford, as well as many, many other things, PharmCat, ClinPGX, CPIC, ClinGen. And so she's a tremendous colleague. And so as soon as we got to work in 2020, we started to kind of think of how could we bring up PGX at Stanford. And so we were very fortunate to identify a kind of a funding mechanism called Catalyst a few years ago. And that was a new program to fund kind of high innovation ideas. Typically, they want to see a product to like spin out into a company, but they'll also fund programs as well too. And so long story short, we were able to get high-level kind of funding and visibility from this program to enable us to kind of bring up PGX at Stanford. And so we had a lot of back and forth as to what exactly are we doing, because everybody had their own ideas. But in terms of trying to get this accomplished, we had to go through multiple levels of scrutiny from the institution as well too. And so these are kind of the two big domains that we always pitch to the hospital leadership when it comes to like pushing on these innovative kind of programs, especially in PGX, is to what's the benefit of this? And so there's a landmark paper came out in the Lancet in 2023. It was a clinical trial in Europe using a 12-gene panel and about 7 countries. This showed a 30% reduction in adverse drug reactions when implementing a PGX panel in kind of a pragmatic trial design. In addition to the safety and the efficacy benefit, the big one that the hospital administrators also react to is the potential for cumulative cost savings for the institution. And unfortunately, this is really, really hard to measure at most medical centers, given the complexity of how these larger institutions can manage it. But some of the data that has come out is from like smaller, more focused, combined payer healthcare systems where they can actually track these important metrics. This was out of Kentucky at a small group there that showed after a PGX panel intervention in 2018, they actually had the ability to monitor overall healthcare costs. And this is a huge task to be able to do that. But what they were able to show, of course, you don't see anything the next day. PacBio started working with Sarah and Nina and the team on designing a custom PGX panel specifically for HiFi sequencing using enrichment-based capture from Twist. And so Nina and I are writing this up now too, but basically it's a 49 gene panel that's commercially available now at Twist, one of the Alliance panels. And it basically was designed to be kind of clinical, but also translational research content. So it has all of the genes that have CPIC guidelines, which are underlined, as well as additional content that's a little bit more on the discovery side or a little bit more on the kind of translational research side as well. And then the emphasis too was to try to do as much full gene sequencing as possible. But some of these genes are so big that that was not possible. But for the vast majority of them, we actually could do full gene sequencing, which has been extremely important for phasing. So now that we had the catalyst support to push on our PGX program, that enabled us to justify bringing in this innovative panel that previously we had been collaborating with outside of Stanford. And so this was kind of all the sequencing was being done at PacBio at that time. But now that we had the resources to do it in-house, that prompted us to bring it all in and build up a whole new informatics workflow for clinical HiFi enrichment sequencing, in addition to the genome work that we were doing already at that time. But I do want to pause and just mention a challenge with pharmacogenomics, and again, thanks to Mike and his team, is that taking the raw data and translating that to the actual PGX nomenclature is not trivial, especially when you're sequencing at scale. We used to do this manually at Mount Sinai when we had a genotyping platform, but now that we're doing sequencing, it's just not amenable at all, and it needs an automated solution. And so thankfully, at Stanford, as I mentioned, Terry's group had already built, or has been building, a tool called PharmCat, which actually takes a VCF and turns it around into diplotypes, phenotypes, and also pulls in CPIC recommendations. But it misses CYP2D6 because anything with structural rate rearrangements is not possible to be interrogated by PharmCat. And so thankfully, Matt Holt and Mike's team at PacBio, we've been partnering with them for the last year and a half on StarPhase, and he briefly mentioned it in the previous talk, but our strategy then was to actually bring in both PharmCat as well as StarPhase into our computational pipeline because we want the dosing recommendations from PharmCat, but we need StarPhase to do the translation for CYP2D6 as well as the HLAs as well. So this is a really highly innovative pipeline and workflow. And so once we finally kind of narrowed down what this is going to look like, we spent the last year and a half kind of building this. And it's been a real pleasure, as I mentioned, a huge team science with Terry and Michelle's team. Vishnu is from Catalyst, and he's been the kind of glue between our laboratory staff, our engineers, and of course, there's a humongous amount of work with our IT group at Stanford Health Care, because they also invested in the genomics module at the same time that was also previously mentioned with the Singapore talk in terms of being able to provide best practice alerts at the time of prescribing. So in terms of what our scope of work was, we had the PGX panel from PacBio and Twist. So knowing that it had content that wasn't exactly robust enough for clinical actionability, we spent a little bit of time to kind of whittle down what did we want to validate clinically, and that narrowed it down to 14 genes. However, for the HLAs, we have to kind of put those on pause. This is Melody Zhang. She's the HLA lab director at Stanford. She's been extremely supportive, and a great colleague. So we're doing a little bit of further validation work on the HLAs for now. But in terms of what we're live with now, we validated the 12 genes highlighted in green, as well as specifically went through allele by allele and identified which alleles are functional, clinically valid, and have actual recommendations affiliated with them. So we remove ourselves from identifying things that we don't know what they mean. So in terms of the scope of work, basically what we wanted to do, we call it Spira. We really wanted to build an end-to-end solution that starts with a saliva kit for patients at Stanford Health Care. And then there's kind of 3 products at the end. One of them is a PDF report that our lab's LIMS system can generate. The other one is filing discrete results into Epic Genomics module, that's mentioned before. And then the third one is part of the genomics module functionality, where those discrete results in Epic are then mapped to genomic indicators, and those are mapped to various medications. So downstream, the providers can have these best practice alerts that pop up. However, that's a real simplification, and I really like to show this slide because the amount of work that all of our team did, this is a brand new wet lab procedure for us on the clinical side. We had to set up a new computational environment. I mentioned that at the very beginning for our short reads. We mirrored that for our long read workflow. We used a lot of the best practices from PacBio, and there's Starphase, and then FarmCat. We have archival storage in our Google Cloud, Kubernetes infrastructure, and GIMS is our LIMS system where we drive all of these behaviors. So we have two pipelines. One we call the bioinformatics pipeline. The second we call it the translational pipeline, and then that generates these reports and discrete results that we file by HL7 into Epic. So we're really excited about it. It's been a tremendous amount of work, and so I'll just show a little bit of the data before kind of wrapping up. And so I previously showed our strategy for analytical validation work, right? And so on the genome side, we saw those numbers, but here's just a snapshot of the panel numbers in terms of just analytical accuracy. And so we'll note, though, that the numbers of variants is much smaller, right? Because now we're only looking at 12 to 14 genes, whereas the genome-wide. So we do see some numbers that are a little bit off, but that's just because of the low counts. But overall, over 99% accuracy, recall, and precision for our small variants. And then with our repeatability, we've done these over and over and over again and always come back with really robust, reproducible variant calling. And then, of course, for specimens, we need to have saliva validated. And so, again, really, really great to see paired blood and saliva working really well for these enrichment panel data. And so we also have, this is just the variant calling. That doesn't mean that all of these variants are clinically significant. So we also have the parallel data from FarmCat and from Starphase to show that the actual diplotypes are also consistent, not just the small variant calling. And so in terms of clinical performance, that's when we bring in the translation tools. And I don't want to spend too much time, but this is just the FarmCat results. So we had well over 100 samples of known controls for different PGX haplotypes from all of the genes. And I have the Starphase data that I'll save for next time as well, too, that's looking extremely encouraging. The CYP2D6 loci is so complicated. So any of the discrepancies that we see with CYP2D6, we don't automatically assume Starphase is wrong. It's more likely that the reference was wrong because of the resolution we're now getting with our panel and with the accuracy of Starphase. So we're still kind of needling through some of those unique discordances. But the take-home message was our accuracy and our 95% confidence intervals are really high and tight, respectively. And so this is what launched a week ago for our pilot program. And so I'll just end here in terms of the Catalyst people. They wanted to see an actual product and new workflow, but they also want to see it being used. This is a huge component of the program, is to actually partner with some of our clinical colleagues. And this was decided to be with a genetic counselor, a pharmacist, and primary care, or specifically senior care. So we have a pre-test counseling visit with a genetic counselor who screens patients who might be eligible for the panel. And then when the lab results come out, there is a post-test counseling pharmacist visit to go through all the medications that the patient might be on, as well as all of the different results from the panel. And thankfully, we're partnered with some implementation science colleagues at Stanford, because the goal here is just to do a successful pilot, but the high-level goal is to expand across multiple service lines across SHC. And so we really need to build in soft metrics as to what does success look like in terms of this program. And so we're happy to report that that just came out or just launched last week. So we'll be sequencing our first patients in the next day or two. So I'll end there. So I just wanted to mention and summarize that, long-read amplicon sequencing enables really highly accurate and phased variant detection across clinically significant genes. I don't think there's anybody who would debate that. And then we're thrilled with the genome sequencing performance. It's accurate, it's greater sensitivity across all these other challenging regions. And as the data is coming out, obviously, with the ability to see more comes the ability to have an increased diagnostic rate, which is ultimately what's most important. And then finally, it's been a real journey in terms of our PGX work with PacBio, but it's just extremely gratifying to be able to now launch a clinical, highly innovative enrichment long-read PGX panel with the team across multiple groups at Stanford, as well as with the group at PacBio and Twist, and this is all being supported by Catalyst. So again, thanks for the opportunity to be here. It's a huge group of collaborators and colleagues. Our team is made up of clinical lab, informatics, and interpretation, but we have a whole bunch of other colleagues and operational management to thank as well too. So apologies for going a little bit over time, but again, thanks very much. There's my email there if anybody wants to contact me afterwards.